So Tommy asked me to talk about the history of the discovery, understanding of face cells. And since I was present during at least some of that history, I put my own personal perspective on it. Just to put it in context, when Charlie Gross began the study of the temporal cortex, virtually all the work in the visual system up to that point had been done in V1, almost all of it in anesthetized cats. The experimentation was more, I would say, almost clinical, in that Yubo and Wiesel would study cells with edges and slits and so on, and just basically just describe verbally what happened with a few examples sprinkled in to illustrate what things went on during the experiment. In fact, there was a history of V1 written by by Richard Jung in Germany, who had before Yubo and Wiesel had tried to study V1, but they had a machine that only swept horizontal and vertical bars across the machine. It was a very nice piece of German engineering. And he said, but because they were limited to horizontal and vertical edges and weren't just waving things around on a screen, they missed the orientation specificity of V1. In fact, most V1 cells weren't responsive at all. And they thought, well, this is what it should be, because why would anyone think that if you stuck an electrode into the visual cortex, anything sensible should happen? It just should be thinking, and processing in a way that wouldn't make any sense. But then Yubo and Wiesel came in, and then they described how they could drive cells with oriented edges, and then it was very influential, them describing you could go from oriented edges to corners. And people quickly ran with that idea and thinking about maybe that was the path at which you would build up complex representations, sort of stick figures of complex forms. But although people from anatomical work understood that the temporal cortex would be sort of a logical either endpoint or peak of the hierarchy for this process, that there was nothing known about it. And so when Charlie, initially here with Peter Schiller, but then later on his own at Princeton, started studying the temporal cortex, it was a great surprise to the field what he found, which was that cells had visual responses, that you could stimulate cells, and they had receptive fields. All very surprising that this should happen so far into the system that it should be that cell properties might be straightforward in any way. But then in the 1972 paper on the IT cells, they had this figure, which was the most shocking figure, I think, in visual neuroscience to that point, which was their illustration of the order of stimulus preferences for what they call the monkey hand cell. And so the cell that would respond weakly to the circle, but then as the image got closer and closer to the image of a monkey paw, the response would get larger and larger and larger. And again, you notice there's no data here. It's just the picture of the stimuli. But that fit with what people did at the time, which was that you would kind of wave these things around and you just report what happened. And part of, I think, the controversy was that you just had to take people's word for it, that this is what happened. And of course, then you have to think, and so there was a tremendous weight put on whether the person was believable or not. They actually know how to do an experiment, and there was a lot of talk in the field about the people you could or couldn't trust at the time, and so on. But this had a very big effect. Now, this paper is often cited as the paper that reported face cells, but I think that's essentially incorrect. Where it comes up is this sentence. And it said that for at least 3 IT units, complex colored patterns, e.g. photographs of faces and trees, were more effective than the standard stimuli, but the critical features of these stimuli were never determined. So from my experience at the time, all the talk was about the monkey hand cell, nothing having to do with faces, because there was nothing ever illustrated about faces.
Why Peter Schiller does not appear as a co-author in those papers?
There was a very early paper. So this paper was a Journal of Neurophysiology paper, which had even things like the monkey hand pictures in it. There was an earlier science paper that was back in those days you would publish a science paper and then you would publish something in Journal of Neurophysiology that actually had the data in it. And so that was what they did here. So there was that science paper in '69, but there was an even earlier paper, I think maybe '65 or something, in which they just basically got evoked responses from IT, and that's the one that Peter is on, because that was done here. And then Charlie went off to Princeton, and then there's continued separate sets of experiments. But Peter and Charlie are on the original paper.
I found funny or interesting that Peter, even in the year 1990, did not believe in face cells.
Well, he wasn't with Charlie when the work was done. I was all at Princeton. Oh, well, so at the time, again, it was the thing then that wasn't about face cells. It was all about the hand cell. And if you read people accounts like Blakemore and Horace Barlow talking about how they imagined visual recognition to work in the brain, they all dealt with the significance of the hand cell and dismissed the idea that you would build up cells that would respond to just one thing. And it was all because of the theoretical argument, well, there's just too many things. And so it can't work that way. And since it can't work that way, really, what do we have here? We have Charlie Gross saying that this was the order of preference of a cell, right? And so it wasn't very strong counter evidence. Then I became a graduate student in Charlie's lab. Oh, this is the issue that the issue that was debated was whether you would have individual cells responsive to individual objects, and so it was widely discussed at the time. Actually, Charlie, in his description of the history, says that the phrase grandmother cell came from Jerry Letvin here at MIT, who used that phrase in a course. But there were no grandmothers described at that time, although Jerry Letvin had talked about bug detectors in the frog tectum, which sort of fit that idea. And I would say that the weight of thinking in the field was, rather than having cells represent grandmothers, that it might be that you'd have cells, again, the part-based type models, you'd have cells that respond to parts, and then basically from kits of parts you can put together anything, faces, tables, and what have you. So then, in the course of my thesis work in Charlie's lab, we came across this area that was actually outside of, just beyond the border of traditional IT cortex in the upper bank of the superior temporal sulcus, which we called the superior temporal polysensory area in an earlier paper. And then, and there the cells had really, really complex properties. And with a postdoc who was in the lab at the time, Charlie Bruce, we thought we would actually study the response properties of cells in that area. And these were examples of penetrations from that studies in the STS. And there we did find cells that really did seem highly selective for faces. And by then, we had developed what we called the semichronic preparation. All this work was done in anesthetized animals. And until we developed the semichronic preparation, where you could recover the animal and then record from the animal again, these things were always done as marathon recording sessions. This is the way Cuba and Weasel did it. You'd record from an animal for 2 or 3 days until either the experimenter or the animal dropped dead. So if you found an interesting cell, there wasn't an awful lot of time to set up any kind of experiment. And so you had to sort of can things. But since we could go back in the same animals over and over again, and we realized that there were cells that responded to faces, we could prepare some stimuli in advance to use. And then this is an example. This is what nearly everybody was doing at the time. I brought an example of the printout of of a chymograph camera is you would photograph the oscilloscope face, and then you would use examples. And so this was considered a lot better than just saying what the cell did. That you could, and this is what Yubo and Wiesel did. I mean, after all, they were doing it. Is that you could show the response of the cell, an example response to the different stimuli, and we were able to do things like drop the eyes and do scrambling and so on. So I would say that in the visual neuroscience community, that this one figure in that paper had attracted a lot of attention. I think I've told the story before. We were visited in Charlie's lab by a very, very prominent Japanese scientist whose name, unfortunately, I don't remember, but he had said that this one figure was the most famous neuroscience figure in all of Japan, and I was like blown away. This is fantastic. I said, so in Japan, do people really believe this? And he said, oh, no. And that was it. This is people just dismissed it because, again, it was it didn't fit this idea that this visual system shouldn't work this way. And again, you're almost just taking someone's word for it that this is actually what happened. And I put, where did I have the, I put Charlie's picture in there. Where was Charlie? Oh, there. I wanted to show Charlie Bruce, who was the postdoc. This is the lab at that time, and this was Charlie Bruce, who was the postdoc at the time, and there's Charlie Gross at the back. And then noticing we were all holding toilet brushes, which at that time was, in Charlie's lab, was considered the perfect visual stimulus, and I brought one of the original toilet. This was one of the most widely used toilet brushes from that time because you could get them in all different colors, and they had a straight edge if you were in an early visual area, but if you were in an area like IT, you could turn it around and stimulate with the fuzzy, complicated edge. And it would seem like it was the perfect visual stimulus. And when Ricardo Gattas was in the lab mapping all the extrastriate visual areas, it was almost all done with toilet brushes. It was really unrecognized, the value of the work. Anyhow, so we had published that paper, and then the next paper that came out on the face cells came from Edmund Rolls and Dave Parrott. And this is another case where at least they had quantitative data in the sense that they showed bar graphs of cells' responses to different things. And they did have also manipulations like, they take out the eyes and the ears and so on, and then show you what the response would be. But this is one of these sort of sociological issues that basically hardly anything that Edmund Rolls said at that time did anyone believe. And so again, it was easy to dismiss the studies. And the other irony at the time is that Edmund's study was done in awake animals, and the awake animal work was actually almost the most untrusted because it was the least controlled of the situations. And so there was very, very poor control over where the animal was looking. There's an animal looking around the room. You have experimenters waving around stuff. And I remember very clearly Richard Morris telling me that he had visited Dave Parrott's lab at the time, and he was a monkey sitting in a chair, and they were recording from his cortex, and there were people swinging on ropes in front, like Tarzan, in front of the monkey, and they were recording responses. So that was like a typical story that came out of Parrott and Rolls. And then, the conditions got better. So what happened was computers got to be used more and more, although Yubo and Wiesel never, ever adopted the computerized controlled experiments and all that. It was all clinical testing. But it started to, as we moved into the modern age, that we would have computers, they'd do histograms, you'd have interleaved trials, and so on and so on. It'd be data would be collected under controlled conditions. So we published this paper in 1984 that was based partly on work that was data that was collected at Princeton, in part when I went to the NIH, where we had sort of more quantitative studies of the IT cells. And there were cells that just responded to complex things. That was the most common. It didn't make any sense. They weren't ethologically meaningful. and we did. There was yet another hand cell that was found in that study. But the only thing that people actually remember out of that study was that there are all these the face cells that we reported in that study. And a lot of the things that we did back then were the issues that came up repeatedly in studies after that, which is to what extent was the orientation of the head important? And there were some cells that responded to heads looking forward and other cells that responded to heads looking to the side. And then to what extent were there component features that were critical? So in other words, was it the eyes or was it the mouth or was it actually nothing in particular? It's just the configuration, and this was an example of a sort of configuration cell. And there was the toilet brush. And actually, I here at MIT, I was in the elevator once, and there were a couple graduate students talking about the earlier studies, and I guess they had come across this paper, and one of them said, "You're not going to believe what was actually in the paper." And they said there was actually a picture of a toilet brush in the published paper. So that was presumably what they were referring to, the perfect visual stimulus. Again, so there are things like so this was an example of a cell that responded to profiles of faces, it was all just playing around with different kinds of stimuli. Was it the outline or the features inside and so on? The thing that Charlie emphasized at the time, and I think was we all talked about, was that the cells acted like little people, and so that you can say, like, well, if a thing looked like a face, then the cell should respond to it. That was your best prediction for what a cell would do. If we thought it looked like whatever, then the cell should. But only with this one qualification, that sometimes the head had to be pointing to the side, or sometimes the head had to be pointing to the front. But they acted like little people, and again, that was at the time, although we had published more, I guess you'd say, controlled data. It was, I think it was just disbelieved by the large majority of the community. I know when I went to the NIH and the lab, Mort Mishkin's lab, got reviewed for the first time since I was there. They bring in an outside panel to review, and I was assigned a person who reviewed my case, and he said, "If you continue this work, it will be the end of your career." And he was really serious, and I took it seriously too, and I never did anything in this again. But if you were reporting stuff like this, the feeling was that you weren't a careful scientist. This couldn't work this way, and therefore you had no future doing this kind of work. And the practical problem was that because you had no way of localizing these cells in advance, you just had to stumble across them. And so you might think, oh, if I got a face cell now, I might not get another one for another 5 years. And so there's not the fMRI to identify the face patches. So it was practically, it wasn't very, didn't make a lot of sense to study face cells. There's a lot of sensitive orientation and so on. And then what you had over the years was just lots of studies, again, where people would stumble across cells that looked face selective. They tried to do experiments. It was very hard because of this issue. You couldn't keep going back to find the same cells. And then I think with the paper with Doris that had, with the fMRI localization of the patches, that totally transformed the physiology of face cell, that field. And anyhow, and it's changed radically since then. There you go. That's the history.